Today, we're going to talk about the book that Koloman Rath and I have published a couple of weeks ago. The title is Viroids and Viruses, or Viroids and Virusoids, Nature's Own mRNA, Understanding Viroids, Hepatitis C, and Ending a Deadly Infection, which is the infection by hepatitis C. The two authors are shown in the back here. There is Kolo, and Kolo is on the talk and will be more than happy if there's a question to welcome you. I'm just going to start to read parts of this book. But before I begin that, why would I ever write, or why would we look at the viroids and virusoids? First of all, they cause disease, and they're in plants, animals, and in humans. They're alive as anything. They replicate, they hop from species to species, and they change slightly over time. There's a lot of them, and people don't really understand that. And now that messenger RNA is a drug, particularly with the COVID vaccines, now that CRISPR is a new gene therapy, now that CRISPR plus RNA are being used for cancer therapies, it's really important to understand what RNA can do. And here you have little pieces of RNA, and I'll read from the introduction of the book, little pieces of RNA, maybe the smallest living things, that can go from one species to another, cause disease. They have to transmit from one species to another. They have to get into the cell across a cell wall or a cell membrane. And as you know, if you know about trees or plants, cell walls of plants are much tougher to get across. They have to get across. They have to get inside the cytoplasm. They have to transit to the nucleus, where they go through a very elaborate replication process. Now, almost all of us who are interested in biology know that enzymes are proteins, but enzymes can be RNA. And these small little bits of RNA have enzymes that help them replicate. They cut the RNA and they splice it back together. All of them have that, except a very few that use cellular enzymes to do that. But basically, these little bits of RNA have a lot of information. Not only that, they are compatible with the mechanisms in the cell. RNA doesn't just drift into a nucleus, it is bound to proteins which carry it across. So these little bits of RNA are adapted to our cells in very interesting ways, things we can learn for vaccines and for other kinds of RNA drugs. Things don't just get into the nucleus, I go through a nuclear pore, which is very selective of what it lets in. As you know, all our cells have a nucleus and a cytoplasm, and these little RNAs adapt to that mechanism and get into the nucleus. Once they're in the nucleus, they do something else that's just totally remarkable, which we can learn a lot from messenger RNAs and drugs, which is they copy themselves. They have to have the right signals, so they recognize the cellular replication enzymes. All those are very complicated signals. And as one of the people listening, Roberto Petrarca, knows, one of the things we study is the interaction between proteins and RNA. It's really complicated. RNA folds up just as intricately as proteins do. And it's the shape as well as the content that matters to those exact interactions with the replication. Now, if that were remarkable enough, they do something very surprising. Normally, your nucleus uses the enzymes that's in the nucleus to take DNA and make more DNA. And that more DNA then makes the new cell. Well, that's a replication process which goes on all the time in our bodies. But these little bits of RNA subvert that. They capture the very enzymes, the polymerases, and some of them capture 3 different kinds of polymerase 1, 2, and 3. And capture it for their own ends to replicate their own RNA. So it's using a DNA directed DNA polymerase to take an RNA and make more RNA. Completely amazing that they can do that. If that weren't enough, they have to get out of the nucleus. Getting out of the nucleus is no trick. One of the things that I discovered when I was a professor at Harvard is how intricate the process is of getting RNA out of the nucleus. It's just as complicated as getting something in. There's special proteins, there's blocks all the way along the way. These little bits of RNA overcome all those blocks. Wouldn't that be useful for the new generation of drugs? If that weren't enough, some of them, the virusoids, make their own proteins. And they make them, since they're so small, they make them in a very interesting way. Sometimes they make multiple proteins from the same little bit of nucleic acid. Now, as you all know, the genetic code is written in threes. If you start it at position 1 and read straight through, you make one protein. You start at position 2, you make another protein. You start at position 3, you make another protein. Now, usually there's stop codons in starting with position 1 or position 2, but in this case, there aren't necessarily. So that's the first thing they can do. They can make multiple proteins from the same bit of nucleic acid. And if that weren't enough, some of them very likely use the reverse strand. So while you're making proteins going this way, you copy the RNA that way, and you can make another protein. And some of the most important viruses for humanity, like HIV, actually reads in the opposite direction. Roberto Petrarca and I are looking at that question for SARS-CoV-2 and some others. That is almost the end of the story. The RNA then has to transit out of the cytoplasm, across the cell membrane, across the cell wall, through the plant, and then get into another plant. All of that with a minimal of information. These are really worth looking at. We should study them. And I can tell you that almost all the people I know, in fact, had the CEOs of companies who are working on mRNA drugs, don't know that viroids even exist. Or if they do, it's really vague. They're not prepared to understand what you can do with a little bit of information. That's why this is an important book. It's important for today. It's important for people who want to think about the origin of life. It's important for people who understand DNA chemistry. It's important for people who want to know RNA enzymes. It's important as a tool to understand intracellular trafficking and DNA replication. Not to mention pathogenesis, how these things cause a disease that they do. This is really fascinating stuff. And I'm very fortunate that Koloman Rath has been with me on this journey to see what the latest is about this. And that's what this book does. It is an introduction written for people who are not necessarily specialists in molecular biology or enzymology. This is something everybody should read to know. If you're curious about life, if you want to know what the future of medicine is going to be, if you're actually a drug designer, you want to understand these. And I can tell you, we don't really understand how these are doing all the things they're doing. We have some inkling of it, and that inkling is given in this book. But I would say there's a good 10 or 20 or 30 years of work of some of our very best biologists to try to understand this and use that information to make a whole new generation of drugs.